So next one is cilia and flagella. Cilia and flagella are structures which are concerned with the locomotion in various types of organisms, both in plants and also in the animals. So this type of what the difference we find between cilia and flagella means, generally the flagella are long and their number will be less, either one or very few number will be present in the in the structure where they are existing. But when it is a cilia, the size is small and the number is very high. And flagella will be showing one type of movement where the pushing of water, that means one flagella, if more than one flagella are there, the movement, the activity will be independent of each other, while the flagella will be showing the synchronous or simultaneous pushing of the waters, so that the cell or the organism will be moving front. So likewise the type of deviation can be seen, but basically the structure will be similar with very small differences, or majority of the structure in the cilia and flagella same. So in this one we can see the cilia, flagella or cilia or locomotive structures in the sperm cells or the male gametes of some of the algae, some fungi, can also be seen in the bryophytes. All the bryophytes, majority of the bryophytes we can say, and majority of the pteridophytes. Apart from this one, flagella can be seen in the unicellular locomotive organisms like in Euglena, and cilia are a tuft of structures which are present throughout the surface of the paramecium. Likewise, not only in the reproductive structures, but we can also see the presence of cilia and flagella on the vegetative structures or the body of the particular unicellular organisms. Flagella can be seen not only in the eukaryotes, but also been seen in the prokaryotic organisms like in bacteria. Based on that one, the bacteria are differentiated into, like we have seen, atrichus, monotrichus, lophotrichus, cephalotrichus, peritrichus, like amphitrichus, like different types we have seen. So likewise cilia are concerned with not only the movement of the body, not only the movement of the unicellular organism, but also the movement of the sexual organs or the male gametes to reach the female gametes. Okay, in the structure of flagella, basically basal body is present, or we can call it as basal granule or blepharoplast. Second one, rootlets. Third one, basal plate, and fourth one, shaft. So these are the four structures that we can see, but basically we say that basal body, the hook like structure will be present, and above that one axoneme are present, that is long filament like structure will be present. But here we can differentiate the parts into four parts here. So if you see the structure of the flagella in the vertical manner, so this forms the cytoplasmic level. The center we can see the hub like structure, and on the peripheral side we can see the ABC structures. Here this ABC structures are extending, A and B structures are extending up to the axoneme part. Okay, this is a cytoplasmic level. So this is a shaft, and in the center we can see the core region. Then the radial spokes will be there. So this is a basal body from this region to this part. That means this region, this part is called as a basal body. So this plate like organization is there, no, this is called as a basal plate. And these are the rootlets. So this is a central core region, and these are the, so three sub parts will be there. This is A, this part is B, and this one is the C. So here if you see the shaft region, only in the basal body we can see A, B and C subparts, but here we can see only A and B regions in the shaft. So on this side also A, B and C parts here are present, but here also in the shaft part we can see only A and B. So likewise these are called as spokes. These radial elongated structures which are running parallel are called as the spokes. So this is a vertical section where we can see four parts related to the flagella structure. The first one is basal body, basal granule or the blepharoplast. This is the structure which is present in the cytoplasmic content just below the plasma membrane. Or we can say this is a place where the part where the origin of flagella or cilia will be taking place. That part is called as a basal granule, basal body or the blepharoplast. And for the formation of this one, around this basal granule part, the ATP is very much present, because for that for the movement of this flagella or cilia they require the energy. That energy is present surrounding the basal granule, which is provided for the movement of the cilia. Then the second part is called as rootlet. Rootlet is the part which is present below the basal body that we have seen in the diagram. So which is present into the cytoplasmic content and which is providing some sort of strength to the basal granule. That part is called as a rootlet, which is made up of some sort of tubular structures, which proteinaceous structures. Then the third part is called as a basal plate. Basal plate is present above the basal body, at plasma membrane, at cytoplasmic level, or the plasma membrane where the cytoplasmic content is ended and the plasma membrane is starting. At that level basal plate is present. So above the basal body at the plasma membrane level. Then the fourth one is called as a shaft, which is the part which is protruding outside, which contains a fluidous structure inside this one, and here which contains the filamentous structures, which is made up of a protein called as tubulin. As like our entire cilia and flagella is made up of a tubulin protein. So here this central part is called as a axoneme in the shaft. The central part is called as a axoneme, and here the shaft may be either smooth or it may contain some sort of hair-like structures. Based on that one, we call the flagella as whiplash flagella, and otherwise tensile flagella. Whiplash flagella are smooth in its surface. Smooth outer surface is present without any outer appendages, while tensile flagella will contain flimmers, or we can say that some sort of small hair-like structures are present, so which are assisting the flagella in the movement of the organism by showing some sort of pushing of the waters. This type of variation can be seen in the shaft region of the whiplash flagella and the tensile flagella. If you take the transverse section of the flagella, basically as we said that this is made up of a protein called as tubulin protein. Almost all the cilia part and the flagella part is made up of tubulin protein, and it contains an central structure which is showing 9+2 arrangement. So, what is this 9+2 arrangement? Which actually there contains microfibrils. So, here 9 peripheral fibrils are present and 2 central fibrils are present. 9 peripheral fibrils and 2 central fibrils are present. This type of organization can be seen in the eukaryotic flagella. But in some of them we can see exceptional where 9 peripheral fibrils are present, but only 1 central fibril is present, that is called as 9+1 arrangement. But there are some cases where central fibrils are completely absent. If the central fibrils are completely absent, we call them as a 9+0 arrangement. So, this can be mostly be seen in the prokaryotic flagellas. 9+0 arrangement we call it as. So, this type of variation can be seen in the arrangement or presence of the central fibrils and the peripheral fibrils in the flagella of different types of organisms, particularly in the eukaryotic and the prokaryotic organisms. Here the central fibrils are present. These central fibrils are attached to the peripheral fibrils, and these peripheral fibrils contains duplets, or we can call them as a dimer like structures. In the transverse section of flagella or cilia, we can see 2 central fibrils. So, these central fibrils contains 13 protofilaments in them. They are made up of 13 protofilaments, and these are connected with double bridge like structure. This is a bridge which is connecting both the 2 central filaments, and they are covered with some membranous structure. The protein sheet we consider it as, the protein sheet is present. So, these are central fibrils. This is the double bridge which is connecting both of them. Then here each of them, as I said, the central fibrils are made up of 13 protofilaments, and these are having some connecting spokes. So, on the peripheral side we can see 2 microtubular tubules. So, those are called as A and B microtubules. A and B microtubules are present on the peripheral side. A contains likewise 9 peripheral structures are present here. Actually, A is smaller and B is little bit bigger. So, this is A microtubule and the B microtubule. So, these are A is having a complete 13 protofilaments, whereas B is not having complete because it is partially sharing the wall, and each of A is having 2 hook like structures. A is having hook like structures. The outer hook like structure and the lower hook like structure. Here the lower hook is connected, A part of lower hook is connected to the B of its adjacent peripheral microfibril. Likewise, inner hook-like structure is connected to the B microtubule of the peripheral microfibrils, and each of them is having the transition structure. This transition structure is the one which is connecting the central fibril with the peripheral fibrils. So total 9 transition structures will be there, which are helping in attachment of the peripheral fibrils with the central fibril. And on that we can see radial spoke, which is connected to the A microtubule, but basal side is having little bit swollen structure. And this spoke is connected to the this radial spoke is connected to the A microtubule present as a part of the peripheral microfilaments. So likewise, total 2 central fibrils and the 9 peripheral fibrils which are connected with the spokes, and this part is a transition site which is connecting the central fibril with the peripheral fibril with the help of the spoke like structure, and this entire structure is covered with some sort of membranous lipoproteinaceous nature. So this type of entire structure can be observed in the transverse section of the flagella. Here in this diagram we can see only A and B microtubules. This type of structure can be seen only in the shaft part of the cilia or flagella, but if you take the transverse section of the basal granule, where we can see along with A and B we can also see the presence of C microtubule also, where partially the wall of the A and B microtubules are shared with each of them. That is the reason why the 13 complete protofilaments are not present in B and C, whereas A is having all the 13 protofilaments. So likewise, they have the side chain structures also. The microtubules may contain some sort of the side chains which are assisting in the movement of the flagella. So this type of organization can be seen in the transverse section of cilia and flagella. And which type of locomotion is observed, or which type of movement does the cilia or flagella showing for the movement of the body of a living organism. Here as we are saying, which type of movement, actually here flagella will show simple undulation movements, where when a flagella is present which is attached to a body, the waves will be formed simple like this. This type of simple waves will be formed where the simple movement can be observed. But sometimes the movement is will show the reverse stroke here sometimes. So here the movement will be like this. This is a flagella structure. Where along with this type of movement we can see this type of pushing of the water to the backside, and in the reverse stroke, so we can see that movement of the water in the reverse direction. When the flagella is moving like this, it is pushing the water backside, so that this is the one rotation movement, and here when it in the reverse stroke, so it will push the water towards that side, so that water will be coming in this direction. So likewise, two types of strokes can be seen in the flagellar organization when the reverse stroke is observed. But in the simple undulations, only the water will be moving in the form of a wave-like structure, so little pushing of the water can be observed. What about in cilia? Which type of movement can be observed in the cilia? Cilia, as we said that many cilia are present, so it's a simultaneous type of or we can say coordination type of movement or pushing of the water will be observed. So here all of them are synchronous type of, that means simultaneous or coordinated type of movement where the water is simultaneously pushed by one flagella, one cilia, and the same water is pushed by its back one. Likewise, it will be pushing the water in the same direction. Likewise, the body or the unicellular organ will be moving towards the front side and pushing the water to the back side. And the same way, not only for the movement. For example, if you take the paramecium like organisms, the cilia are not only helping in the movement of the body, but they will help in the engulfing of the food materials or engulfing of the water into the cytostome and cytopharynx like structures. So that means cilia are not only concerned with the locomotion, but also engulfing of the water and also the food particles into the body. And also the pushing out of the waste materials or the undigested materials from the body is also been assisted to some extent with the help of the cilia. And for all these processes, the movement of cilia and flagella is taking place, as we are saying, but for that process they need some sort of, it's an active process. So that means they require some sort of energy. That energy is provided by ATP, which is present just below the basal granule, which is always assisting providing the energy for the movement of the flagella and the cilia.